Thank you very much for the introduction. It is indeed, very interesting how we are bringing together all these omics approaches and are beyond the genomics, and how much they help us now to understand also the genomic discoveries and go further, a step further. So I would welcome our first speaker, and we can have the questions at the end. Thank you.
Tanya?
Should I go first?
I guess
Tanya, it's over to you.
Sure.
Yes.
Please.
Sure. So, hi, I'm Tanya Ward. I'm Director of Bioinformatics at Diversigen. Diversigen is a company that specializes in microbiome profiling, amongst other things. And my background is primarily in a really comprehensive mix of both working with molecular biology in the lab and a clinical research setting, and combining that with the bioinformatics analysis on the back end. And so today, one thing that I wanted to talk to you about is a recent study I was involved with in looking at a multi-omics approach for IBS. So IBS is a pretty complicated disease type. So we've got a lot of recurrent abdominal pain and discomfort, changes in stool frequency and form. But basically one of the reasons we classify someone having IBS is that they don't actually fit into any of the other IBD categories. So we really don't have a good understanding in terms of what IBS is, in terms of what's happening in the body, and how to prevent it and cure it. The other challenging part about IBS is that there are periods of remission. So subjects who have IBS can have really bad symptoms for quite some time, and then go through a long period of remission, making things like cross-sectional study design very difficult. Because you never know when you take a sample from an individual, whether or not it's really going to represent, a true snapshot of a longitudinal component of their disease. So one of the goals that we had with this study is to integrate a multi-omics approach with longitudinal tracking to try and identify IBS subtype-specific mechanisms. And so how we accomplished this is we had a cohort of patients with irritable bowel syndrome with diarrhea, irritable bowel syndrome with constipation, and age-matched, gender-matched, BMI-matched healthy controls. From those individuals, we sampled stool longitudinally once a month for 7 months. We characterized that with metagenomics, so shotgun sequencing, and metabolomics. We also captured the dietary information for these individuals. And then at the baseline and the 7th month component, we also had biopsies taken via colonoscopy. And so from those biopsies, we were able to do more microbiome profiling via 16S, more metabolomics profiling. We also were able to do host transcriptomics, host methylome, and a cytokine panel. We also got the metabolomics and cytokine panel for serum samples that were taken both at the beginning and last time point as well. So when we think about integrating omics, I think I've covered every single omic possible, except for maybe the actual S&P array. But after reviewers' comments, we actually went back and did that as well. So we're looking at a whole encompassing approach in terms of tracking the IBS mechanisms. So I'll just give a brief overview in terms of what we found. And this was published in Cell in 2020, so I encourage you to look up the paper. But we started with the metabolomics. And what we were able to find is that individuals with IBS had much higher levels of something called hypoxanthine. And hypoxanthine is involved in the purine recycling pathway. So intestinal epithelial cells have a really hard time creating their own purines. So they have to use the salvage pathway. And what we found is people with IBS had much lower levels of this particular hypoxanthine. When we then looked into the shotgun data, so looking at the microbes in the stool samples, we were able to see that the function, so that the genes or enzymes specific for the degradation of hypoxanthine were much higher, again, in individuals with IBS as opposed to healthy controls. So it started to help us build this story in terms of we have individuals with IBS, they have much lower levels of this metabolite in their stool. their microbes in their stool are actually able to degrade that metabolite much faster. But then we're still missing the host component. So is this affecting the intestinal epithelial cells? Are they reacting in any way to this that are giving some of these IBS phenotypes? So what we did is we looked at that transcriptome data. So of the biopsies, we found that the other host side of hypoxanthine degradation is also impacted. So a gene called PNP, which is involved in the purine nucleotide phosphorylase, and also the xanthine dehydrogenase, which is also in the host genome. Those are actually upregulated in terms of transcription in the host epithelial cells. And that PNP level, so the gene involved in degradation in the host, directly correlates with the level of hypoxanthine seen in the stool. So building on that, we're basically able to generate this new model of IBS mechanism, which is we have hypoxanthine, which is much lower in individuals with IBS. And it's in part due to both the host and the microbes in the GI tract. So we have changes in the transcription related to genes in the host that are involved in the degradation of hypoxanthine. And also we have an upregulation in those genes in the microbes, which are also eating up that hypoxanthine. So that also gives us new drug targets, right? So we can either target things in the microbiome to prevent those microbes from degrading hypoxanthine and leaving more for the host. And we also have targets in the host. So we could potentially increase purine in individuals with IBS to potentially supplement that pathway and deal with that decreased loss of hypoxanthine. Or we can also target the host cells specifically in terms of their PNP regulation. So the take-home message of this case study, and I know it's a lot to take in with just chatting through it, is basically by combining all of these multi-omic techniques, we are able to really get at what is causing differences in different components of this pathway and resolving that pathway in terms of its role in IBS in general. And if it weren't for combining those features together, we wouldn't be able to kind of piece everything together. So that's kind of my case study for Multiomics. And I'll leave it there for a moderator.
That's very interesting, and it's a very interesting disease also to study in the multiomics, and having the possibility of getting stool samples have actually given you a lot of rich data to look at. And I mean, I'm not sure if we are having questions at the end, but maybe we can have a few now and then take the discussion from them. And one I see which is relevant is what are the capabilities of the DNA genotech tubes in terms of collecting the multi-omic samples? Would you like to maybe explain a little bit on that?
Yeah, sure. So DNA genotech has some great stabilization technology. So when we think, and especially in this realm of COVID, where we don't have access to patients and healthy cohorts readily in a clinical setting, being able to do sampling at home is really important. So being able to get a stool sample, stabilize it, have it mail ready so that you can ship it out, makes things a lot easier. So if we think about DNA genotech tubes, we have the ability to do that in terms of the microbiome profiling, so for shotgun sequencing or 16S profiling. But we also have tubes that are stable for metabolomic profiling. So I think it's up to 5 days we can actually stabilize that, so we can do the metabolomics as well. So that's a big advancement because metabolomics in this study was really the jumping off point to point us in the right directions. And without being able to collect that data, we wouldn't have necessarily been able to piece this together at all.
And the stool samples are can be preserved not on the fridge.
Yes,
they can
exactly.
stay. They are stable.
Yeah,
very important. And then I guess if someone would not have the capability of having, a lot of multi-omics in their study, the question is, if they were to choose one, what would you go for?
So I think that really depends on what the the model is that you're studying, right? So if you know that something, your system is directly related to, if it's IBS, obviously you're going to target something related to the GI tract. So whether that's the shotgun profiling or the metabolite profiling of stool samples. My company, since we focus on microbial profiling, is probably going to kill me for this, but personally, I'm seeing a lot of promise in the metabolite profiling. And in this particular case study, like I mentioned, it was really kind of the smoking gun that helps us out.
I got to say that's music to my ears. Yeah. Sorry, I didn't mean to interrupt. I'll just introduce myself briefly because that's music to my ears. Hi, I'm Greg Michelotti. I'm the scientific director at Metabolon, where we're a company that's done metabolomic profiling for the last 20 years and developed expertise in the field and are one of the leaders in metabolomic service provision in this regard, and have an extensive library of molecules that we're able to report on all biological samples. So it's not just serum or stool, but basically if a sample has biochemicals in it, we're able to extract it and analyze it. And really everything Tanya just delineated there in that exemplar study really shows the power of a multi-omics approach in deconvoluting complex diseases. And to the moderator's question in terms of what omics approach might be best, echoing what Tanya said, it depends on the study and the disease. If there are clear genetic drivers of an illness, then it makes sense to understand the genetics behind that. But I do like to point out that even when we understand the genes or the proteins that might be involved, there's such a level of complexity on that end of the central dogma of molecular biology that we really don't understand the deep ontologies behind genes and proteins, giving their bewildering array of complexity that's surrounding them in terms of mutations and splicing and post-translational modifications and subcellular localization. It really confounds our ability to understand and interpret the changes in those omics, and especially in the context of complex diseases. And you really need a multi-omics approach to do this. And where we focused at Metabolon is on metabolomics, because they are proximal to the phenotype. They're the tip of the spear, if you will, in terms of proximity to the phenotype. And they integrate all that upstream information to provide a comprehensive view for what's going on. So in the example that Tanya provided, it really helped identify the actual mediators that might be involved in the process and give insights into drug design. So it's really a multi-omics approach into doing that. And it's not just, the exemplar example of gut dysbiosis in a gut-related illness like IBS is really just the beginning of what this multi-omic approach and metabolomics could do to help deconvolute this. Because we've done a lot of work, and pardon my filibuster here, but we've done a lot of work to use metabolomics and leverage metabolomics to understand the upstream omics, such that, for example, host genetics, that we understand still a very small percentage of the genes in the genome, and not even talking about microRNAs and other things that are there that we really don't have a good understanding for. And we've used our technology to functionally map variants of unknown significance in genes of unknown function. And there's a number of papers in this regard that are out there that metabolomics has been used to functionally map genes of unknown function, so that we've added to the knowledge base the function of over 1,000 genes using this approach in various systems, and by extension in proteomics as well. And then when you think of metagenomics, although metagenomic profiling is extremely useful for understanding the composition of a particular microbial community, it doesn't provide yet the functional impact. Even though there's some programs, software programs like Human, that can try to get there, there's still functional gaps in knowledge of understanding the metagenomic information and how that results in functional outputs. And the microbiome is unique in that it's really soluble mediators that are used to communicate with the host. And that's where metabolomics is particularly useful and powerful, because we classify and identify and quantify those soluble mediators involved in microbial metabolism and how they affect the host. So it's really the integration of all these omics, and with the onset of metagenomics and the power of metagenomics, to really understand complex diseases. And just to take the IBS example that Tanya talked about further, it's not just in the gut. Like gut dysbiosis doesn't stay localized in the gut, but these soluble mediators can actually enter the circulation and affect all organ systems and affect outcomes in all diseases, even in the brain where they can cross the blood-brain barrier. And there's a report that we published recently showing that there is a microbiome component to amyotrophic lateral sclerosis, or ALS, a neurodegenerative disorder, where soluble mediators from specific strains in the gut enter the circulation, enter the brain, cross the blood-brain barrier, and influence disease outcomes and the course of disease. So it's really systemic illnesses that the gut can, that microbiome and microbial activity can influence. And it's this multiomics approach. And with regards to the microbiome, really the power of metabolomics to leverage and identify the soluble mediators in identifying those diseases. So sorry for that long-winded discussion, but I think it's important to say that it really is, these are systemic issues that really benefit from multiomics approaches, and you really need everything together.
Exactly. I mean, I agree. I couldn't agree more, because I've worked a lot with the metabolomics as well. But when we actually go and link those to the genes and link those to the proteomics, then we go further into the pathways and we can translate the information we get from metabolomics much better and much more efficiently. So, Craig, anything else from your end, or shall we move to Nima as well?
Yeah, no, I'm excited to have Nima talking, because he kind of puts it all together from the analysis standpoint. So I'm excited to hear him speak as well.
Thank you,
Hi,
Nima.
hello,
Over to you.
All right. Yeah, I'm an assistant professor in Stanford. We are a lab of artificial intelligence and machine learning people primarily. We work on the underlying mathematical problems that need to be resolved in order to integrate multiple omics datasets. Our lab is mostly disease agnostic. We work on a broad range of diseases. We collaborate broadly with translational scientists. But the theme that is common between most of our studies is a multi-omics theme. We often have multiple assays applied to the same patients. This could be proteomics, transcriptomics, single-cell assays, imaging modalities, even EHR data sets. And then we try to put all of these together. One lesson that we learned pretty early on is that while there are specific machine learning algorithms developed for each of these omics assays, none of them can be effectively used to put all of these data sets together. Part of that is because these omics datasets have different sizes, they have a different number of biological measurements available, but things actually get even more complicated than that, because these datasets have different modularities and different internal correlation structures. For example, if you have a large untargeted metabolomics dataset, that assay will be measuring a lot of different things, but there will be a lot of overlap because it's measuring the same pathways over and over again at different endpoints. In contrast, if you have a targeted proteomics dataset, if that has been targeted correctly, it's probably measuring different pathways rather than overlapping pathways. So the machine learning algorithms that we are developing are specifically designed to be able to first understand the characteristics of each omics dataset, and then to effectively combine them to increase overall predictive power and to reveal mechanistic connections across different omics datasets within the context of that particular disease. Again, because these priorities change from one disease to another, we have seen metabolomics assays work really well in some settings and poorly in other settings, again, depending on the disease state and the biology that we are going after. So we try to take an agnostic approach, use a broad number of assays that we think are relevant, and then let the machine learning algorithm figure out what actually works, what doesn't, and which connections exist in that particular disease. I think I can stop here and answer questions.
Yes, that's again very interesting, and the data dimensionality issue, especially on the untargeted metabolomics data, is something always very difficult to deal with. Have you worked with try to matching metabolomics data across different platforms?
Yes, that is quite interesting. We have worked on trying to matching targeted and untargeted, and also we have tried to match across different biospecimen, match across plasma and urine, for example. These are all pretty interesting problems that are not straightforward.
What would you say are the common pitfalls in this analysis that you would maybe guard, not, I don't know, explain to someone now, so in order not to for them to go into these pitfalls when analyzing this kind of data?
Yeah, so there are two things. When designing the study itself, I would say sample overlap is very important. We often get studies that have a lot of missing data in one assay and then a lot of missing data in the assay. Before you know it, sample overlap across 7 different omics data sets becomes 0. You have 0 patients that have everything, and that dramatically reduces a statistical power. The other thing to be careful about is that you can't just take off the shelf machine learning algorithms and throw the data at them. Again, because these data sets have different characteristics beyond just size, also modularity and correlation structure, the underlying assumptions of those machine learning algorithms often get violated. You have to design algorithms that are aware of the context of each omics data set before trying to integrate everything.
Thank you. Would anyone else have something to add, maybe?
Really, it's just a question. In terms of your approaches, Nima, to that, are there any modalities that are particularly more difficult to work with to integrate into a multi-omics approach? In other words, so people use genomics a lot, and you have all sorts of statistical approaches that you can use from that, Mendelian randomization, et cetera. But in terms of, is that the anchor modality that you want to build everything around, or what are your thoughts about the challenges of bringing these together and what best ways to do that?
Yeah, we don't really anchor everything around a specific modality unless there is a good reason to do that. If the disease states suggest that it's a genetic based disease, then of course you can do that. But generally we don't, we treat everything equally, and we let the machine learning algorithm figure out how to put everything together. In terms of your first question, that is something more difficult than others. In my experience, you can debate whether some of these qualify as omics data sets or not, but imaging-based assays often are harder to integrate with other assays that just come in a tabular format, because especially if you're trying to use some kind of deep learning to automatically extract features from them. If you're looking at a translational study with a limited number of patients in it, automatically extracting features from an imaging data set becomes a little bit difficult. But if you are extracting features from the images in a fairly traditional way, even that wouldn't be too bad.
Thank you.
Thank you very much, Nima. I was also thinking that a lot of these machine learning techniques are mostly used for prediction, sort of to predict the outcome. But a lot of the questions we try to answer with Multiomics have to do with causality and causal inference. Are you analyzing both of those questions? Do you adapt your machine learning algorithms to sort of causal inference?
Yeah, so we have a number of algorithms that are under development in the lab for extracting causal connections from multi-omics data sets. Of course, feel free to send me an email if you like to see examples of that. We don't have anything published yet, but that is a pretty interesting problem, and it's not something that you can do using machine learning in isolation. For truly inferring causality, you need at least an element of time, preferably an element of an intervention that targets that causal link that you're interested in. And when you put those together with the right causality inference algorithms, you can do a pretty good job with inferring connections between modalities.
Nina,
Thank
I
you.
think you kind of
And
alluded to this. Sorry, can I ask a question real quick? So, Nima, I think you alluded to this before, but at Diversigen, we do a lot of like custom work. So, in example of this multi-omics data set, putting different omes together, whether it's related to the microbiome or not, and dealing with that complex analysis and trying to bring those feature sets together. We've noticed that it's really difficult to have any sort of standalone solution. Like it seems like every single project is really custom in the approach that we have to take due to things like missing data that you mentioned, the types of omics data we're trying to integrate. Do you feel like we'll ever get to a state where we could have kind of like standalone tools that, you know, individuals could potentially plug and play with, or do we think things will always be really custom?
No, that's what my lab works on. We are trying to take tools that are only working here and there and figure out what is common between them, figure out what are the right components, and how to have the machine learning algorithm itself learn to adjust itself to new settings rather than a human being having to do that every time. For some problems, it's easier than others. I'm pretty sure we have figured it out for predictive modeling. We now have algorithms that can work pretty well, regardless of what the multiomics data set is. For other problems like causal inference, it's a little bit harder, but that's what we are working on, and hopefully we'll get there in the next few years.
So do you have an idea, and this is for Tanya and Nima too, because we have an idea about this, but I was wondering what you may think in terms of what kind of power we're talking about to support these machine learning approaches and how to bring these data together to find causal inferences. So as we start looking more and more at complex diseases, IBS, and how that can be stratified, cardiovascular disease, neurogenic disorders, so everything's getting to be these complex diseases, and the low-hanging fruit, so to speak, is rapidly disappearing. What kind of power is needed to develop and leverage these approaches?
So as you probably, go ahead.
No, go ahead, Nima.
All right. Sorry about that. As you remember from very early math in primary school, probably if you have two observations and two unknowns, you can solve that system. But if you have more unknowns than you have observations for, you can't really solve the system. But the way that translational research has developed is quite different from that, in that now we have these multi-omics data sets with hundreds of thousands of features, unknowns that we need to model, and only a few hundred patients. So that's quite divergent from what traditional mathematics would expect. So now we have sparse models and all kinds of machine learning techniques that try to solve a complex problem with lots of features and very few patients provided. So, but just by definition, the problem is a difficult one, and it's one that has many correct solutions. You can't just arrive at one solution. So if you ask the statistician in me, how many patients do you want for a multi-omics data set with 50,000 features? My answer is going to be in the millions of patients in terms of statistical power. But that's not going to happen in reality, so we have to get a little bit creative. We have algorithms now that are able to integrate prior knowledge into the machine learning algorithms, and that helps leverage knowledge that's already in the public literature to increase predictive power. That's just one of the tricks. But then the sparse models and other techniques also help us reduce the number of patients that are needed. In practice, we can usually get away with a few hundred patients per condition if you're trying to do some kind of causality analysis. If it's just a predictive modeling study, it would depend on your effect size and the disease that you're trying to predict, but again, usually in the hundreds.
Yeah, I would totally agree with what Nima has said. I like that if you have 50,000 features, you should have 1 million people. that so hits the nail on the head. I would also add, just in terms of when you think about effect size and when you're designing your studies, Another thing to consider is the longitudinal versus like cross-sectional nature of studying your system. So if you're working on something that's dynamic, like the microbiome, potential metabolite profiling, things can change from day to day, from week to week, from month to month. So that's something that you should account for in terms of initial study design, if possible. You can get more power as well from looking over time with individuals and account for kind of that noise that happens with snapshots. So just another take on it.
Okay. And that's a great point, because we are expanding our ability to, so we're working with genetic epidemiologists and working with very large cohorts. So we are starting to get to those very large studies now. We're not getting to millions yet, but the very large population studies are in the tens of thousands now, and growing as populations and programmatic efforts to understand complex diseases gain strength. I think we're going to be able to change the calculus of having the number of observations versus the number of variables changing, and they'll be tipping it at some point in the future. We're not there yet, but in the future. But for now, a longitudinal study design, like Tanya was saying, where you have repeated measures, where your studies can derive additional strength from repeated measures within individuals, is something that we've seen. It's a very powerful approach.
I mean, that's definitely a strength, and will help us understand also, the variability of these measurements that have to do, with time or with inter-individual variability. But it is true that studies get larger, and we're going to hit the millions at some point. But still, I think a lot of the omics data would come from bringing studies together. So, and I wanted to bring that to our discussion. I mean, how can we bring different studies together, having measured omics, maybe sometimes with different ways, but also even with the same platforms? Maybe some comments on that. How can we borrow from each study, and that how can we use those to replicate the results or increase our power doing, meta-analysis?
Yeah, that's a great point. And so I'll start with that, because metabolomics is unique in that as a data stream, you have to pay attention to specific aspects of the data generation and the technology, so that missingness is treated in a very specific way, and data can be batched and harmonized in a very specific way, given the LCMS technology that we don't have time to get into today. But we've thought about this a lot, and for at least metabolomics, we have an approach of being able to run data sets longitudinally and being able to merge them based on quality controls and anchor samples that we have across the entire run, across all the samples that we run, so that you are able to harmonize data sets, assuming that study designs can be appropriate, because you can't just take willy-nilly disparate study designs and just slam them together. There has to be careful design in terms of how samples are collected and analyzed, and what the demographic of the population is, so you avoid any potential confounding that can happen due to the way samples are collected and technical aspects of things. So we paid a lot of attention to being able to do that, and so that you can take, and in fact, now this is becoming programmatic, because the Transomics program through NHLBI or TopMed is now bringing this together from a multi-omics perspective. So national samples from different populations and different cohorts, bringing these together in a multi-omics way to understand complex diseases that are focused NHLBI. So these are approaches that are being supported programmatically and then being developed by investigators therein. And so at Metabolon, we thought a lot about that and are able to do that in terms of stitching data sets together to support multi-omics approaches.
Yeah,
that's a great question.
And I was thinking, as you said, that the actual multiomics could help this harmonization of data across. I mean, even looking, for example, at the genetic determinants of metabolites between different platforms could tell us that they are actually measuring the same metabolite, and whether we can combine them in a way. Any other comments on this? Or
I would say that in terms of, I can really only comment in terms of batching things together from the metagenomic standpoint, but we've done a lot of that in terms of pulling data and doing large meta-analyses across multiple data sets. I think from our end, it's a lot easier to correct for batch effects across different studies. As long as we, like Greg mentioned, know what happened upstream, we can usually correct for it and bring that signal back out so that we can have a nice harmonized data set across the board. So that is fairly well understood in the field of metagenomics, but I know that that might not be the case in the other OMS.
Yeah. It could be more difficult in other omics, but still, I think we are going there. We are trying to find ways, as Greg says, to bring data together, and that would be very important.
Yeah.
Any other comments on this? I was also thinking that we discussed about these large biobanks and the fact that we will have a lot of omics data to work with in large numbers, but at the same time, a lot of people hearing us will thinking of how can they design their own study. So could we discuss a little bit the study designs and for preclinical or human studies and the actually sample sizes that we are looking for maybe?
I can start with that, and certainly welcome the thoughts of the panel. I know we've looked at this very carefully for metabolomics and understand what the variation is in metabolites and to identify biologically relevant signals from the studies that we've analyzed over the years, both preclinical and human. And when you start with preclinical studies, usually in genetically identical backgrounds like black 6 mice, you have decreased power because you have very well controlled conditions and a controlled genetic background. So very little inter-subject individual changes, as opposed to humans, where there is a great deal, as you mentioned, Ivana, that there's a great deal of individual variability. So you need higher power for those. So we typically see studies in the 6 to 10 range that are relevant for the very well powered for identifying metabolomic changes in preclinical studies. And the higher you get up that phylogenetic scale in terms of complexity, the more samples you need, so that we've actually seen very, very clean, well-powered studies in the 25 to 50 sample human range per group. Now, these are samples per group of a given condition that you're looking at. So you have a control versus some sort of whatever the metric may be for the illness in the groups, so that at 25 to 50 samples per group in humans, you have a very well-powered study to identify biologically relevant signals and changes. And obviously, you have to go higher and higher. So the rarer the metabolite and the less represented is in the population, the higher you have to go into the power. But that gives a very broad overview in terms of what we see in metabolomics.
Amazing. We have few seconds left, so I would welcome any concluding comments from our speakers. I would like to say that this session gives us a flavor of the power of these multi-omic approaches and how much more we could go with sort of epidemiological studies and human studies into mechanisms and pathways that before we thought we could only look in them with basic research, but now we have the power to go into this level of details, which is very promising.
Yeah, I'd just like to say one thing very briefly, that the technology is just astounding today, that we have an ability now to look at multi-omics, genes, proteins, metabolites in ways we haven't before, and it derives from the ways that we can collect samples as well. Tanya mentioned that there's an ability now to collect stool to look at both genes and metabolites that is stable for a long period of time, and we've worked with her and her group to develop that, make sure that it's validated for metabolomics. So now you have an ability for not just clinical collection, but field collection of samples, and be able to analyze things in very large ways and with a high degree of power to identify these biologically relevant signals in the population and deconvolute these complex diseases that underpin and understand why people respond differently to diseases or drugs. And it's because of the technology and our way to collect samples for those analyses that ultimately go to NEMA's door.
Thank you very much, Craig. I would Okay,
Well,
thank you all very much. Our
Thank you.
time, fortunately, is finishing. So thank you very much for the opportunity, and best wishes to everyone.
Thank
Thank you
you.
all. Thank you to the organizers.
Bye bye.